Good to see you again. Next section we're going to talk about deals with cell life cycle. In this section we'll talk about the development of cells within your body. We'll also talk about a clinical application associated with this. So for our first concept map, our main concept of that map, we'll start on the cell life cycle. And for bubble A, we know that cells have two major periods, and these periods define the process through which the cell develops. So bubble A, cells have two major periods. Bubble B in our concept map, one of those periods is described as interphase, and this is an important component of the process of the cell growing. And when the cell grows, the cell carries on metabolic processes, so processes that provide the resources, the energy, allowing for the cell to grow. So bubble B, interphase, cell grows and cell carries on metabolic processes. Bubble C, when we're talking about bubble C, we're talking about cell division. All right, so the process through which the cell actually divides. All right, so in addition to cell division in bubble C, I would put this is the process where the cell replicates itself. Okay, in bubble D, when we further describe cell division, we know that one of those functions is to produce more cells for growth in the repair process. As we've talked about before, cells are one of the foundational components of all living things. They're one of the foundational components for levels of organization. So within cell division, you have a function that is geared towards producing more cells for growth and repairing. Okay, for our next concept map, the main concept of that map should be titled DNA replication. So within DNA replication, this is associated to what from a DNA standpoint is occurring when cells start to divide, when cells start to grow. So we know bubble A, we're talking about genetic material being duplicated, and when this occurs, it readies a cell for division into two cells. So it's important that that genetic info be duplicated so that the required material from a genetic standpoint can go into the next cell. So bubble A, genetic material is duplicated and readies a cell for division into two cells. Bubble B, we know this occurs towards the end of interphase, which is a topic we just described in our last concept map. So that involves during the cell growing and during that process through which the cells carry on metabolic processes. So bubble B, it occurs towards the end of interphase. Bubble C, we know in this particular relationship when DNA is replicated that DNA uncoils and each side serves as a template. And if you look at this diagram to the right, we can see that relationship. And I hope you can see this. I moved the box that records what you can see to the top left corner, but if not, we'll go over again when we get into class. So what you have in our DNA, you have the old template strand. And as you can see, if you remember from organic compounds, a component of DNA involves nucleotides. And these nucleotides are defined by various components. So within that strand, you have your old template, and it has our nucleotides associated with it. And then on the opposite strand, the one that's been synthesized or created, you have the new nucleotides, which create the combinations that make you and I unique from a DNA standpoint. Okay, so main concept for our third concept map involves events of cell division. So what events have to take place, or what events do take place that promote the division and that drive the division of those cells? Well, the first of those, bubble A, involves mitosis. We talked about the nucleus as being one of the central components of all cells, so mitosis is the division of the nucleus. So for bubble A, we know that one of those events involves mitosis, which is the division of the nucleus. Bubble B, we know this results in the formation of two daughter nuclei. And we should remember what nuclei are from our nucleus section. So bubble A and B deal with mitosis and the specifics associated with that. So when we're talking about mitosis in bubble A, we're talking about the division of the nucleus. And when that nucleus divides, you have the formation of two daughter nuclei. Bubble C, cytokinesis. Now, if we think of cyto, another term that would resemble that would be cytoplasm. So when we're talking about cytokinesis, we're talking of the division of the cytoplasm. Excuse me. If you remember, the cytoplasm is what holds the organelles, the metabolic machinery that drive the cell. So for bubble C, cytokinesis, it's the division of the cytoplasm. Okay. The second component of cytokinesis, bubble D, we know that begins when mitosis is near completion, and it results in the formation of two daughter cells. So for this information, we'll start with this Monday. We'll review it briefly and then apply it to a situation, and we'll go from there. If you have any questions prior to class on Monday, let me know. Thank you.